So, hello everyone. This is going to be the next lecture for your histology. And today we are going to discuss about the histology of eye. You might have studied it from different sources, maybe YouTube lecture, or maybe you might have studied books like D. Fiore's Atlas of Histology. So you might already have, the basic concept, whether from your anatomy classes or your high school, that what are the parts of eyeball, what is sclera, what is cornea and everything. Even though you might remember or you might have forgotten, I will give you a brief description of everything. I will start with the basic anatomy, then I will go to the histology. In this lecture, I might not be discussing the entire histology of the eye, but primarily I will be focusing on the sclera and the cornea. So let's start with the basic anatomy of eye. Our eyeball is spherical, and to be exact, this is oblate, spheroid. When we see the eyeball, we see the outermost layer of the eyeball. Here, this part is your sclera. This part is your cornea. Well, I just highlighted cornea, but can you see the cornea? The answer is no. Cornea is transparent. So what you are seeing here, the green color, is actually your iris. And what is cornea? It's a anterior bulging. When you close your eyes, try to touch the eyelid, and you will see there is a bulging. That bulging is your cornea. It's a transparent layer. Then you have your middle layer, which is uvea. Then you have internal layer, which is your fossa. So let's start with the layers of the eyeball. Layers of eyeball. So we have three layers. First one is the outermost tunica. Tunica means layer. Fibrosa. Because this layer is rich in collagen fiber, that is why we call it fibrous layer. This comprises of anteriorly 1/6 cornea. Let's take the photograph. This part that you see here, this is your cornea. And the second part, which is the posterior 5/6, is your sclera. The white colored structure that I just showed you in the beginning of the lecture. This entire part is your sclera. And the point where the sclera and cornea meets, this is known as limbus. Got it? Or the corneoscleral junction. Now comes the middle layer, which is very rich in blood supply. This is known as uvea, the middle layer, which is tunica vasculosa, because this has highly blood supply. This is tunica vasculosa, also known as uvea. Uvea comprises of choroid posteriorly. You see this part? This is your choroid. You see, outermost you have white color layer, the innermost is your yellow color layer, and in the between you have a brown colored layer. That is your choroid. That is your posterior part. And the anteriorly you have ciliary body and iris. And the ciliary body holds lens. Let me show you. The continuation of the middle layer or the choroid anteriorly. This is your ciliary body. This is your ciliary body. Ciliary body gives rise to fibers. These are known as suspensory ligaments or the jonular fibers. What does it do? It holds your lens. Got it? This was anterior layer or the anterior part of your uvea. You have one more anterior part that is known as your iris. This part that you see here, this is known as iris, which is this green part. So we Indians have a brown iris, and other people most can have the green iris or the blue iris. So iris can be of different color, and the color depends upon the type of melanin, the distribution and the concentration of melanin. So we discussed outermost layer, the middle layer, and comes the innermost layer. This layer is known as tunica nervosa or the nervous tunic. Why? Because this has retina. You see this yellow colored layer which is forming the optic nerve. This is your innermost layer or the tunica nervosa. So this was the basic, the layers of the eyeball. Outermost layer, middle and the innermost layer. Today we will be discussing the histology of the tunica fibrosa or the anterior 1/6 of your cornea. We discussed, where it is present and everything. So let's discuss the histology. Suppose this is our sclera. This is our cornea. This part was our limbus. When we go 3 millimeters to our limbus, one layer covers your eyeball. This is known as Tennant's capsule. Why am I telling you what is Tennant's capsule? See, Tennant's capsule actually separate your eyeball from the periorbital fat. Your eyeball is present inside the orbit, and in the orbit you have periorbital fat. So it separates your eyeball from the periorbital fat. Now, why it is so significant? Because this is the structure which provides the site to attachment of your extraocular muscles. Tennant's capsule provides this attachment to your extraocular muscle. For example, your rectus superior, medial rectus, and all of those extraocular muscle. You see this? Extraocular muscle. So let's go to the histology of your sclera. We will take everything slowly so that we don't mug things up. So just give some time, have patience, and we will understand every single thing. Comprises the 5/6 posterior portion of tunica fibrosa, white in appearance. Why white? Because this is made up of dense, irregular connective tissue, and this is rich in collagen fibers. Got it? This is made up of dense irregular connective tissue, which is very which is, you can say it like this is structurally very rigid. So it provides protection to eyeball. This provides site to attachment of extraocular muscle. In the anatomy of eyeball, you will study you have rectus muscles and the oblique muscles. So they provide the site to attachment, the Tennant's capsule. So this was our sclera. If we see the layers of your sclera, we have 3 layers. We have episclera, which is the outermost. This is highly vascular because if you study the anatomy of the eyeball, you have short posterior ciliary artery and anterior ciliary artery in this layer, which form 2 plexus, the superficial plexus and the deep plexus. So this one is the outermost layer and this is highly vascular layer. The second one is known as sclera proper. This is avascular. But where does it receive the blood supply from? The deep plexus supplies the blood here. And then there is the innermost layer known as lamina fusca. This is also avascular. Then what supplies the blood? Below the sclera you have choroid, and choroid is also very vascular. So blood vessels from the choroid will supply what? Lamina fusca. So 3 layers of your sclera: the episclera, the sclera proper, the lamina fusca, and the function. It has a function of protection so that it provides the rigidity, it provides the site to the attachment. It is white in appearance. Now we saw the sclera. Now let's move to the cornea. If any one of you might have gone to an ophthalmologist, You might know what is slit lamp biomicroscopy. What we do is we take a light bulb, we pass it through chamber, and in the chamber there is only one small opening, the vertical opening. What it does is all the light which comes from here now will be moving in a beam of vertical light, and when it falls onto your eyeball it gives it this appearance. You see, this is the vertical beam of light. Why am I telling you this? Because if you zoom in, you can see the histology of your cornea. I mean, I'm not kidding. You can see here you have the different layers of the cornea. White colored appearance that you see here, that is stratified squamous non-keratinized epithelium. And this entire part, the middle part that you see here, that is nothing but your cortex. And on the inside you have your endothelium. Well, I'm not, I won't stop here. In fact, we will be going to the slides. But if you, know these things and, you connect it clinically, you understand it better. So cornea. Cornea comprises the anterior 1/6 of tunica fibrosa. This is also made up of collagen fiber. But here, collagen fibers are type 1 and they are regular connective tissue, unlike the irregular connective tissue. This is avascular. I mean, if it doesn't have blood supply, how does the cornea receives gases like oxygen, the glucose as nutrient and everything? So you might have studied there is something known as aqueous humor. So non-pigmented epithelium of the ciliary process forms this aqueous humor, and it goes from here posterior chamber to the anterior chamber, and from here it supplies your cornea. So this is avascular layer of your fibrous layer. And why do we need it to be avascular? Just think, this is your eyeball and this is cornea. If it had blood supply, it would be red, right? And tell me one thing, which is good, if the cornea is transparent or cornea is red? If the cornea was red, we might have had opacity there and we might not have seen everything clearly. Just think like you have your camera and you put a red marker over it. You will see the red markers in your photograph. So it's very important for the cornea, the lens, all of the refractive structures to be transparent. And that is why during the development of the cornea, it gets avascular. So avascular, made up of collagen fiber type 1, anterior 1/6 of tunica fibrosa. In the eyeball, to perceive the light, we do something known as refraction of the light. We have lens, but surprisingly, 75% of our light is refracted by cornea. Why? Because see, even though cornea might not have high refractive index, but cornea have curved structure. Because of this curvature, this provides 75% of your refraction. And those who want to remember, the refractive index is 1.376. You don't need to, but if you want. And refractive power of around 43 to 44 diopter. That is your cornea. Now let's look about the layers of cornea. First I will tell you about the layer, then I will show you it on your slide. First of all, if this is cornea, we will be going from outside to inside. First layer of cornea is made up of stratified squamous non-keratinized epithelium. What does it mean? Stratified, it means this is multiple layered. Squamous, it means the outer layer cells are flat. Non-keratinized, it means they are not producing keratin. And epithelium, it means this is a kind of epithelium whose outer layer is flat and is not producing keratin. This is the outermost layer. If we see this layer, the outermost cells are flat and these flat part have microvilli. Why do it need microvilli? There are two things. First of all, in your environment you have oxygen. Cornea takes oxygen from the environment. That is why those who wear contact lenses, doctor usually recommend, please take it before night. Like when you go to sleep, please take it out. so that your cornea can breathe, because cornea literally takes oxygen from your environment. So if you increase the surface area, that would be better to get the oxygen. Second thing, your lacrimal gland form tear, and if this is flat, tear can come directly down. But if you make the microvilli or disc-like structure, the tear will accumulate here, here, and here. So this will provide sort of lubrication. That is why the outermost layer, I will write it like this: outermost layer, flat cells bear microvilli. Why? To trap tear and exchange of gases. Got it? This was your superficial layer, or the outer layer of this epithelium. Let's draw it with this microvilli. Now comes the middle layer. Middle layer comprises of wing-shaped cells. These are wing-shaped cells. And at the bottom, you have columnar epithelium. Got it? Columnar epithelium is sitting onto the basement membrane. That is why these are also known as basal epithelia. Columnar epithelium. And they have capability of division. Got it? What did I say? Your cornea has 6 layers. The outermost layer is stratified squamous non-keratinized epithelium. This epithelium has further 3 layers. The outermost layer of the flat squamous cells, the middle layer of the wing-shaped cells, and the innermost layer of your columnar epithelium, columnar cells. Columnar cells have the capability of division. Why? Because they sit near the basement membrane. And since this layer has rich blood supply directly from, the sources, not actually, but this is getting indirect blood supply from your aqueous humor, not blood supply, indirect supply of nutrient from the aqueous humor, and sitting onto the basement membrane, this layer has capability of division. This is the first layer. Let me show you here. This is your cornea. If you take it, see it under the microscope, this is the outer layer. How can you say this is outer layer or inner layer? Because this layer is quite thick, and stratified epithelium has thick layer, as the name says, stratified or multiple layered. See this is multiple layered cell, and you see at the basement you have these dark colored structure. That is your columnar epithelium. Let's take another image, this one. Can we appreciate that the outer cells are flat here? These are wing-shaped irregular cells. And at the bottom you can see these are columnar cells. Got it? This is the first layer of your cornea. What is the second layer? The second layer of the cornea is known as, yes, Bowman's membrane. Now see, wherever you see epithelium in the body, an epithelium always has basement membrane. So this is the basement membrane of this stratified squamous non-keratinized epithelium. This is basement membrane of epithelia. But why do we call it Bowman's membrane and not, the basement membrane? The reason is, all of the basement membrane of the body, when we stain it with periodic acid sniff reaction or the PAS stain, it is positive, but this one is negative. That is why we also call the Bowman's membrane as false basement membrane, and thus giving it a separate name, the Bowman's membrane. This was our second layer. Now what is the third layer? The third layer is cortex. In the photograph here, I told you, just look at this image and you will see that this thicker portion was nothing but the cortex. You see this entire part, this is nothing but the cortex. This cortex is very thick, like this comprises 90% of corneal thickness, made up of collagen type, yes, collagen type 1. Anything else? Besides collagen type 1, you don't have to remember. It can have glycans from your biochemistry. You might have studied what are glycoaminoglycans. For example, you will have keratin sulfate. When in ophthalmology, or when you study about the eye, wherever you study, this word kerato. Kerato means not the keratin here. Kerato means cornea. For example, keratitis, that is the inflammation of cornea. So here you have keratin sulfate, and this sulfate is present into your cornea. That is why known as keratin sulfate, type of glycosaminoglycan. That is your third layer. First one was epithelium, second is the Bowman's membrane, the third is the cortex. Now is the fourth layer, but we will study sixth layer first, or the innermost layer, that is endothelium. Now see, this is the innermost layer of the eyeball, not the eyeball, the innermost layer of your cornea, and you have aqueous humor here. Now aqueous humor has the oxygen, the glucose, the amino acids and everything for the inner cells, but in order for it to get inside, this should be thin, right? That is why this endothelium, or the innermost epithelium of your cornea, this is made up of simple squamous epithelium. This is made up of simple squamous epithelium like this. This is the inner side, so aqueous humor. But one thing is, suppose in ophthalmology you might have studied IOP. Aqueous humor generates a pressure over this so that things can transfuse. This IOP can cause the fluid to enter inside, let's say the endothelium, very microscopic structure, very thin. The endothelium can get damaged, fluid start to go inside or maybe between them. That is why the endothelium have tight junction between them, and the tight junction prevents the entry of fluid between them. So again, endothelium is the innermost layer of your cornea, made up of simple squamous epithelium, and it has tight junctions, also known as macular adherens. These tight junctions prevent the entry of aqueous humor inside the cornea. Why? Because if aqueous humor gets inside the cornea, it will make the cornea edematous. Edema will be in the cornea, and then we will not be able to perceive things clearly. That is our innermost layer. Now I told you, wherever you see endothelium or any epithelium always has basement membrane. And the basement membrane, since this is going to be towards the outer side, this is your layer number fifth. This is known as Descemet's membrane. Descemet membrane is nothing but the basement membrane of endothelium. In fact, the endothelium makes the basement membrane or the Descemet membrane. Got it? The fourth layer, this is present before the Descemet membrane. That is why this is known as pre-Descemet's membrane. So earlier we only had 5 layers. We didn't have the pre-Descemet membrane, but we had one Indian scientist. His name was Dr. Dua. He was an ophthalmologist, and he found this layer. That is why we also call it Dua's layer. Got it? So we have 6 layers. This one is stratified squamous epithelium, the non-keratinized stratified squamous epithelium. Just a second. Non-keratinized stratified squamous epithelium, the Bowman's membrane. The inside you can see the endothelium and the basement membrane of the endothelium. This entire part that you can see here, this is the cortex, and here you have pre-Descemet membrane. So stratified squamous epithelium, Bowman's membrane, cortex, pre-Descemet membrane, Descemet membrane, and this layer endothelium. This is your outside environment, And this is your aqueous humor or the anterior chamber. Anterior chamber filled with aqueous humor. And this is the histology of your sclera as well as your cornea. See, this lecture might be slightly longer. I want you to understand things, because if you understand things, you will retain it forever. And you are not studying to pass any exam whatsoever, your university exam or any entrance examination. You might pass it by remembering the answers. But what will you do when you are at clinics? I try to incorporate, multiple things. For example, the slit lamp biomicroscopy, so that you can see the subject in real life. Unfortunately, what we have studied till now, this is just the bookish thing. We can't, relate things from one subject to another subject. So please don't do it. जल्दी का काम शैतान का होता है. Okay, take time, understand things better, but understand it in a way so that it retains with you always. So all the best. I hope you guys might have liked the lecture. If you have any doubt, just text me. I'll be there to answer you. You can Instagram me or you can WhatsApp me, whatever suits you. Have a good day. Goodbye.